Okay, so I guess I couldn't finish it in 3 videos, so here's the finale. Primary active transport we talked about a little bit. We said that it uses the use of energy by the cell. So what you're seeing here in this image is the cell actively moving molecules, in this case sodium ions and potassium ions, and moving sodium out. So notice that sodium is going out, it's being expelled from the cell, and potassium is being brought in, so you can kind of see it on this part. And that's to ensure that there is a difference in the concentration between the inside and the outside for those two ions. But the cell has to use energy in order for that to maintain that, because otherwise things would ultimately kind of get to equilibrium, and equilibrium is not a good state when we're talking about these ion concentrations. So we'll talk a little bit more about why that's true, but the idea here is that either way we're looking at primary active transport. When we look at secondary active transport, I kind of think of it as like piggybacking off of primary, because in primary energy is used, in secondary we're using the electrochemical gradient that was set up by primary. In other words, by that, the primary used energy to create an imbalance of ions on one side of the membrane from the other. And then by allowing that to naturally go back down its concentration gradient, what we get is other things which can move along with that. And that's what we're seeing here. So amino acids and glucose are two materials that typically go into cells this way. So they're doing this kind of a piggyback affair, coming in by using the power of the primary active transport to move in, again, against its concentration gradient. So that's that. Okay, there's one other type, and that's something called bulk transport. So if a cell needs to take a lot of materials in or push a lot of materials out, it also can use energy to do that, and that's called bulk transport. How that happens is several different ways. And the general process of bringing stuff in is called endo. I think endo always sort of somehow means within. And then the cell, here's cyto, has to do with cell. So endocytosis is bringing materials into the cell. And then exporting is exocytosis, so that's helpful. There are 3 types of endocytosis, so that's the one we're going to look at first. One of them is called phagocytosis, which literally means cellular eating. And what that is large particles that normally can't go through any channels, can't diffuse through the membrane, are going to, there will be some receptors on the membrane itself that will help these little particles to attach to the membrane, and then the membrane surrounds it, and then it brings that material in this little vacuole, which is basically a piece of that membrane that was above now surrounds this thing that's coming in. So that's phagocytosis. Phenocytosis is much the same, except for in that case it's usually a little bit of fluid that's coming in. Sometimes some other salts and things come with it. But the same idea. But we're really looking at bringing in some liquids, some fluids. In receptor-mediated endocytosis, this is a little bit more specific yet, and that's that there are specific receptors, usually proteins, that are sitting on the surface of the cell, facing the outside of the cell, and they are very specific to some substrate, something that we're going to bring in. And so when those things, such as these stars that we see above here, they actually, when they're in the presence of this, they bind to these specific receptors. And then having done that, the same idea is that the receptors this whole thing is brought in to the cell. The membrane kind of surrounds it and then pinches off and comes in. And this is called receptor-mediated endocytosis, meaning it's the receptors which are kind of guiding this activity. It wouldn't happen without them. Now, they put in this term clathrin. Clathrin is another piece of this this type of, or another part of what happens when this thing has this vesicle. So you can kind of see that the clathrin is in the same region as these receptors on the outside. So once those come in, now this vesicle that you see here actually has clathrin coating it on the outside. And that's going to help to guide it to certain areas where it's going to need to go. Finally, last thing is that a cell can get rid of things, and that process happens all the time, and it's called exocytosis. And so if the cell produces something, let's say the endomembrane system and the ribosomes make proteins, they send it along the ER, it goes to the Golgi, gets modified, we put some things on it, and now we're going to send it out into the bloodstream, out of the cell. So what happens is the Golgi actually surrounds that substance, whatever, is going to get exported by the membrane again here. But when the membrane then gets to the cell plasma membrane, it easily fuses, as you're going to see in our lab coming up next week. They just kind of melt together, and then that material that was in here then gets basically kind of gets spit out. So that's the exocytosis that we see here. And that's it, at least for the moment. I'll talk about more of this in class.